So hey everyone, welcome to CoreVet. So today in this particular session, we will be starting with the chemotherapy. So in chemotherapy, we will be taking first two lecture would be of the general chemotherapy, and after that I will be starting with the classes of the drugs, continuing from the sulfonamide itself. Okay, so starting with the chemotherapy. So what does the chemotherapy means? So chemotherapy means the usage of the drugs that are different from the different classes of the drugs that you had studies in your earlier lectures. How so? How these drugs are different? See, you had studied about antihistaminics, right? You had studied about antihistaminics, right? So what does antihistaminic do? What does they do? They inhibit either the histamine secretion or they just block the histamine receptors, right? These two functions are performed by the antihistaminic, right? So what is it's doing? It is particularly what it is doing. It is altering the physiological role of histamine. Means, see, if there isn't surge in the histamine, obviously there would be an allergic response that we can observe in the body, right? So if we are giving the antihistaminic, like levocetirizine you are giving, it is an antihistamine, right? In allergy, we generally give it, right? So what you are doing, you are just suppressing that particular histaminergic response to just confer or to just ensure that the body will reach a normal physiological condition, right? Means earlier, what was there? It was a rise in histamine in the body, right? What it lead to? It lead to allergic reaction, right? Earlier, when there was a rise in the histamine, there was an allergic reaction that you observe. Okay, so when you gave what antihistamine, what it does in decrease in allergic reaction, either by decreasing histamine secretion or by blocking the histamine receptor. Okay, so what it was meant to be? It was meant to just subside or to just reduce the histamine response such that the body come to normal physiological condition, right? To reach a normal physiological condition. So what was the purpose of the antihistamine is that it will lead to a normal physiological condition which was abnormal earlier due to the rise of the histamine. It may be due to any external stimulus which will lead to the rise in the histamine. Okay, so that's why we gave the antihistamine, which will reduce the histamine secretion or it will just block the histaminergic receptors, right? To just confer or to just ensure that body will reach a normal physiological condition such that there will be no allergic response or any other response that will be created by the histamine. Okay, so it was just to ensure that body will remain in the normal condition. Okay, so these classes of drug ensures that the body physiological condition or the natural chemical substance that are there inside the body, they remain in a certain amount such that they do not cause any abnormal reactions in the body. Okay, so the class of the drugs which are the chemotherapy, okay, the class of drugs that comes under the chemotherapy, what they ensure? What was the purpose of the drugs that came under the chemotherapy? We know that the other classes of the drugs like antihistamine, what is the purpose of these drugs? To just confer or to just ensure that the level of the histamine that is there in the body, it is normal such that the body physiological condition is normal. There isn't any abnormal condition that is produced due to the histamine. I'm just taking an example of the classes of the drugs of the histamine. There are several examples we could take over here, but I am just taking about the antihistaminics over here. Okay, so if we talk about the chemotherapy, so the class of the drugs that comes under the chemotherapy, how they differ from the antihistaminic. I'm just taking an example of the antihistaminics. There are several classes of the drugs we can take over here, but I'm just taking an example of antihistaminic over here to such that you can understand what's the difference between the chemotherapy and other classes of the drugs that you had studied earlier. You can also take an example of NSAID over here, which inhibits the PG synthesis, right? The NSAIDs. It will inhibit the COX-1, COX-2, right? You can take an example of the NSAID 2 over here. Okay, so the classes of the drugs that you had studied over here earlier, how they differ from the chemotherapy. See, the chemotherapy, the classes that comes under the chemotherapy, how they affect the body. They affect the body by inhibiting, either they inhibit, okay, or either they kill, okay, kill the microbe. Okay, so see, there are several microbes that are there in the surrounding. Okay, if they enter in the body, they will produce some different reactions, right? So the ultimate etiology will know that yeah, this particular organism is there in the body, which is leading to some certain abnormal condition in the body. Okay, so if there is any certain abnormal condition in the body, so how we are going to counteract that particular abnormal condition? By giving a drug which will either inhibit the function of the microbe or either it will kill the microbe. Okay, but one condition is there. To be a good chemotherapeutic drug, the condition is it should have minimum adverse reaction towards the recipient. Okay, so see an example. This is a microbe. Okay, this microbe entered to body. Okay, obviously it will produce what? It will produce an abnormal condition, right? To counter that particular abnormal condition, what we will do, we will give a drug from a chemotherapeutic class, right? After you give a chemotherapeutic class drug, it will either inhibit, okay, it will either inhibit the metabolic function of that particular bacteria or the microbe that we are considering, or either it will kill that. Okay, such that minimum effect is there in the host. Okay, so this is what the chemotherapeutic drugs are different from the other classes of drugs, from the antihistaminic, NSAIDs, or many others. Okay, so that's how you should understand about the concept, that how the chemotherapeutic classes of the drugs are different from the other classes of drugs that I had showed you with an example over here. That other classes of the drugs are concerned with to just neutralize the condition that is produced by the natural physiological substances inside our body, to just ensure that the body physiological condition could be restored to the normal conditions, right? So here's the definition. So how these drug classes differs from the other classes, that these are designed to inhibit or the kill the infecting organism, which may be the bacteria, the virus, or the fungi, to have a minimum effect on the recipient. So the adverse effect that is going to be produced by that particular chemotherapeutic agent should be minimum. Okay, it shouldn't cause any adverse effect to the particular recipient that you are giving the drug. But in a general, every drug has a certain amount of adverse effect on the recipient. It's just a concern that we write in the definition, yeah, it should have no or minimum effect on the recipient. There are several adverse effects that are there depending on the physiological condition of the particular organism also. It do depend on the physiological condition of the organism also, that what drug we are going to prescribe. That we'll discuss in the second lecture of the general chemotherapy. Okay, so Yeah, these drugs are meant to be treat the systemic infections with the use of the specific drugs which either suppress the infecting organism without the significantly affecting the host. Okay, so yeah, earlier it wasn't considering the malignant cell. The drugs of the malignant cells are now considered under the chemotherapy also. Okay, because the malignant cells are also analogous to the pathogenic organism now. Okay, so the treatment of the neoplastic drugs, okay, neoplastic diseases with the drugs are also termed as now the chemotherapy. Okay, so the treatment that we are making for the neoplastic conditions is by the drugs. Okay, so those classes of the drugs are also considering now under the chemotherapy. Okay, because we are considering that the malignant cells that are there in the body is also in foreign substances, also in foreign organism. We are considering that the malignant cell is also in foreign organism because they are quite an analogous with the pathogenic organism that are there inside the body. Okay, so what are the antibiotics? So these are the substances produced by the microorganism which are selectively going to suppress the growth or the kill the other microorganism at very low concentration. So the antibiotics are the substances that are obtained from the microorganism to counteract the effect or the growth or to the kill other microorganism. That's what the antibiotics are. Okay, so these are these are produced by the microorganism to counteract the effect of the other microorganism at very low concentration that we suspect to. Okay, so what are the chemotherapeutic agents or the antimicrobial agent? So these are either the synthetic or the naturally obtained drugs that are going to attenuate the microorganism, means they are going to weaken the microorganism. Okay, that's what attenuate means, right? So these may be either the synthetic or the natural. Okay, so coming on to the history. So the history of the chemotherapy has been divided into three phases, right? So the very first phase is the period of empirical use, okay, based on the experience they had used. So by the Chinese, the moldy curd was used to treat the boils. In the Hindus, the colmogra oil was used to treat leprosy, and the chenopodium was used to treat the intestinal worms. The mercury was used by the Paracelsus to treat the syphilis. Okay, and the cinchona bark was used to treat fever. These were all based on the empirical. Okay, these were all the treatment was based on the empirical uses, right? Now coming on to the Eldridge phase, which went from the 1890 to 1935. So what the Eldridge seen that the dye stain the microbes. We all know there are several dye that stain the microbes, right? We had done staining in the microbiology also. So what he thought that maybe these dyes that are going to stain that particular microbe, what it may do, it may produce a selective toxicity towards that particular microorganism in higher concentration. So he had used methylene blue, trypan red, etc. dyes to just watch out that whether at certain condition they are selectively toxic to particular bacteria or the microbes or not. Means we know that the microbes are getting stained by a particular dye. We had done that in microbiology. So what we look for is that whether at certain concentration these dyes that are going to stain that particular microbe, are they either selectively toxic or not at certain condition to that particular microbe. That was that what was the Eldridge was thinking. Okay, so what Eldridge did actually, so Eldridge developed an arsenical compound which is etoxyl to treat the sleeping sickness, and Eldridge also developed the S-pheramine and the neo-arsphenamine to treat syphilis. Okay, and what's the two important thing about the Eldridge is that he coined the term the chemotherapy, and he is the father of the chemotherapy that you should remember although. Okay, so the Paul Eldridge gave the term chemotherapy and is also known as the father of the chemotherapy. Okay, so coming on to the modern era. So in the modern era, you should know about the three scientists: the Domac, the Fleming, and the Waxman and his colleagues. Okay, so what the Domgak did, he particularly used the Prontosid, okay, to treat the pyogenic infection. Okay, so the active moiety that was there in the Prontosid was the para-amino benzene sulfonamide. Okay, and the dye portion wasn't essential for the treatment of the pyogenic infections. That it was analyzed later on that the dye portion of the prontosil dye wasn't important for the treatment of the pyogenic infection. The active moiety in the prontosil dye was the para-amino benzene sulfonamide. Okay, and the sulfapyridine was the first sulfonamide which was marketed in the year 1938. Okay, coming on to the Fleming, we all know about the Fleming, that there was a penicillium mold that destroyed the Staphylococcus colony in the culture. Okay, so the Staphylococcus colony was destroyed by the penicillium mold in the culture plate. Okay, and the penicillin was purified by the Chain and Flory in 1939. Okay, coming down to Waxman, he researched on the actinomycetes and discovered streptomycin in the year 1944, and the soil microbes are then proved to be as a treasure house of the antibiotics. That you should know that the soil microbes or the actinomycetes are termed as the treasure house of antibiotics by the Waxman and his colleagues. Okay, now moving forward, so this is something that you may fear of, the classification. It's quite big. I know it's quite big. The classification is big. So how to just remember this particular classification, which is based on the chemical structure? So let me tell you, just skip. I'll say, just skip this particular classification. Why I'm saying like that? To just skip that. Skip this classification because you're going to study all these classes of drugs. You're going to study sulfonamide, you are going to study diaminopyrimidines, you are going to study quinolones, you are going to study macrolides, you are going to study all those classes. Okay, so when you are going to study all those classes, in all those classes you are going to study the several drugs. Like in sulfonamides, you are going to study sulfamethazine, sulfadiazine, sulfathiazoles, all the drugs. So you can automatically write down this classification, that in the sulfonamides, these are the drugs. You can have an example. In diaminopyrimidines, you can write down that trimethoprim is the example, the pyrimethamine is the example. In quinolones, you can write all the examples by yourself. When you see, if you are going to learn the example later on, because you have to study all the classes of the drugs. Okay, you have to study the sulfonamide, you have to study the macrolides. You're going to learn all those examples in the later half. Okay, if you are going to learn the example later, so it doesn't make any concern that you are going to either you learn over here, because you are going to learn more example from there. Okay, we're going to have more example over there in the classes of the drugs, because in the classes of the drugs, the classification are much bigger than this itself, right? So you're going to learn that particular classification from the later part. Okay, so just skip this, and when you will be studying all those classes, when you will be completing the classes of the drugs, you will be automatically going to write this particular classification, which is based on the chemical structure. So just skip that particular classification, which is based on the chemical structure. And because you are going to write this particular classification based on your experience itself when you are completing all the classes of the drug, right? So just moving forward, based on the organism acting on, means we know the microbes are of several categories. It may be bacteria, it may be a fungus, it may be a virus, it may be a protozoa, or it may be a helminth, right? So the drugs that are going to affect the bacterial growth, or it is going to kill the bacteria, they may be penicillin, the aminoglycoside, the macrolides, and the chloramphenicol. They are the antibacterial. If we talk about the antifungal, it may be amphotericin B, it may be graciofluval, or it may be ketoconazole. Okay, talking about the antiviral, it may be zidovudin, acyclovir, amantadine, ramentadine, ganciclovir. These are all the example of the antiviral. Talking about the antiprotozoal, it may be the chloroquine, it may be pyrimethamine, metronidazole, naloxonide. Okay, actually you are going to, when you are going to complete this whole chemotherapy now, when you are going to complete the chemotherapy, okay, when you are going to complete the chemotherapy now, these all classification will be easy for you, because you are going to learn all these example. You can write even more the example that I had written over here. Okay, You are going to learn much more examples that I have written over here in the slide itself, right? So you are going to write down all the classification by your own when you are going to complete all the chemotherapy topic, right? Because you are going to learn several classes of the drug, right? So based on the spectrum, it may be narrow spectrum or the broad spectrum. In the narrow spectrum, you can have penicillin, either you can have streptomycin, you can have erythromycin, you can have tetracycline and chloramphenicol in the case of the broad spectrum. Okay. So based on the type of action, it may be a bacteriostatic or the bactericidal. So the sulfonamide, tetracycline, macrolides, lincosamide, and the ethambutol, all these five are what? The bacteriostatics. These are the examples. There are much more drugs that are bacteriostatic. These are just few examples that I had mentioned over here, which are bacteriostatic. Okay, so if we talk about the bactericidal, the beta-lactam, you can write down beta-lactam directly. The cephalosporin and the penicillin, you can directly write down them as beta-lactam. The aminoglycoside, the polypeptide antibiotics, the vancomycin, the quinolones, the rifampicin, isoniazid, all these are the example of the bactericidal, right? Coming on to the, based on the source from which they are going to be obtained. Either they can be obtained from the actinomycetes, they could be obtained from the fungi, they could be obtained from the bacteria, right? So the penicillin, if we talk about the penicillin, cephalosporin, rhizoflavin, all those have been obtained from the fungi. The polymyxin B, colistin, bacitracin, and the azithromycin all are obtained from the bacteria. The aminoglycoside, tetracycline, macrolides, and the chloramphenicol are obtained from the actinomycetes. You're going to see in the later half, when we are going to discuss all these classes of the drug, you will see that what's the origin of the particular drug is. Okay, you're going to study. Now, this particular classification based on the mode of action is quite important. Why I'm saying like, because you're going to learn about the mode of action here itself, not in the depth, but yeah, having an overview about that, what's the mode of action of a particular drug. See, every drug isn't going to affect the microbe by the same metabolic action. No, they aren't going to just affect the bacteria or the viruses by the same mode of action. There are several mode of actions through which the drug act on a microbe to just have an inhibitory effect on that particular microbe to produce an desirable effect for the recipient. Okay, so the recipient that is going to take a particular drug should have a desirable effect that should be produced. Okay, so there are several mode of action. So first of all, the drug may inhibit the intermediary metabolism. So the sulfonamides is going to inhibit the sulfonamide, the trimethoprim, and the metronidazole. Okay, these are the example of the few drugs that are going to inhibit the metabolic functions. So the sulfonamides and trimethoprim both are responsible for inhibition of the folic acid synthesis, right? You will be going to study that we will be going to study that they are going to inhibit the folic acid synthesis. So that's why they are inhibiting the metabolism. Okay, now there are certain drugs that are going to inhibit the protein synthesis by acting on the ribosomal complex. Okay, so 50S and the 30S ribosomal complex that are formed. So certain drugs that act on the protein synthesis by inhibiting that particular protein synthesis, right? So some examples are chloramphenicol, tetracycline, macrolides, and the lincosamide. Okay, so these classes of drug, what they are going to do, they are going to just inhibit the protein synthesis by having an action on the ribosomal complex. Okay, or either there may be a misreading of the mRNA. Sorry, there may be the misreading of the mRNA also, which is generally done by the aminoglycoside class of drug. Now, there may be leakage of the cellular element. See, if there would be the leakage of the cellular element, obviously the microbe will going to die, right? So the cellular element could be leaked by the usage of the polymyxin B, the nystatin, colistin, amphotericin B, hamycin, and the bacitracin. These are certain examples of the classes of the drug that will help in the destruction of the microbe by what action? By the action of the leakage of the cellular elements, right? There may be drugs that will be going to inhibit the synthesis of the cell wall, okay, by inhibiting the cell wall synthesis. So the classical example is the beta-lactam, okay, then the vancomycin, then the bacitracin, and the cycloserine. Okay, so there maybe the class is a drug that will inhibit the DNA gyrase, okay, enzyme. Okay, so this particular DNA gyrase enzyme is going to be inhibited by the fluoroquinones and ciprofloxacin, maybe the examples. These are the examples. Okay, now talking about the drugs that are going to inhibit the function of the DNA, or the drugs that are going to inhibit the synthesis of the DNA. There is a difference, that some drugs will going to inhibit the function of the DNA, and some drugs are going to inhibit the synthesis of the DNA. Okay, so the rifampicin, both are going to just, what they are going to do, they are going to inhibit the DNA function, while the acyclovir and the zidovudine are going to inhibit the DNA synthesis. There are much more examples that we will discuss while we are going to discuss about the antiviral drugs. Okay, there are several examples for these particular classification itself. But if we talk about out of the classification that we had discussed today, the most important type of classification is this only, that the classification based on the mode of action, and it is quite important, because there are drugs that will inhibit the metabolism, there are drugs that will inhibit the protein synthesis, there are drugs that will inhibit the DNA synthesis, there are drugs that will inhibit the functional aspect of the DNA, there are drugs that will misread the mRNA, there are drugs that will leak the cellular content. Okay, there are drugs that will affect the synthesis of the cell wall. Okay, so that's how this classification is much more of importance than the others. Okay, because other classification you can write by your own. Even this classification, after reading all this chemotherapy, you can write this classification very easily by your own itself. Okay, so this was all about the first lecture of the chemotherapy. We'll be discussing later on about this. Okay, so have a nice day, everyone.